science fans, and welcome to SHE-ensya. Today, we continue our series on genetics and molecular biology by unmasking the technicalities behind genetically modified organisms. So what is a GMO? A GMO is an organism with its genetic content altered intentionally in order to create a difference in its physical characteristics. Please take note, however, that organisms may have their genetic information altered accidentally or intentionally. For example, you could be sunbathing in the beach and your exposure to UV light can actually alter the DNA on your skin. At that point, you become a mutant. Both GMOs and mutants contain alterations in their DNA, but mutations may occur naturally or artificially without intent, while GMOs are always purposely made. Intentional genetic modification does not need to happen in the lab. In fact, as a species, we have been doing it for thousands of years. We've been doing it through the process of selective breeding. But selective breeding is the slow, tedious process of genetic recombination by mating two different organisms with a preferred trait. Selective breeding is one of the reasons why the human species has continued to flourish. Through it, we have created plant and animal varieties that are bigger, have more nutrients, and are also better for our health. Imagine, prior to the rise of agriculture, the lifespan of a typical human is just up to 30 years old, and now we have more than doubled that. Selective breeding has brought about larger, tastier, and most importantly, non-toxic avocados. And without selective breeding, man's best friend would still be man's greatest predator. The process of selective breeding, however, tends to be slow and imprecise. And this is because when you do selective breeding, you have to wait for the natural fertility cycles of the organisms you're working with. This means it can take months or years before you find out if your mating was successful. And despite all of your time, effort, and other investments, the trait that you're looking for might not be expressed in all of the offsprings or even at all.
No! God, please, no! 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 No!
Why is that? This is because the genes that control our physical characteristics occur in pairs, and they may be dominant or recessive. When we say a character or its counterpart gene is dominant, it means that it's a version or an allele that will result to the expression of the physical characteristic whether or not its partner is just like itself or something different. For example, there's a gene for brown eyes and a gene for blue eyes. The brown version or the brown allele is dominant over the blue version or the blue allele. The female in this pair has brown eyes because the dominant brown allele masks the blue allele that she has. What do you think is the color of the eyes of the male of the pair? If you answered brown, then you're correct. And this is because the male also has the dominant brown allele despite having the recessive blue allele. Now, of the 4 possible combinations of their genes, 3 would result to brown eyes. And only 1 would result to blue. The combination that results to blue is the only instance when the recessive trait is expressed. And this is because recessive genes are only expressed when they are partnered with the exact same recessive allele. So, when we do selective breeding, sometimes we don't know if the trait that we prefer is dominant or recessive. And even if we do, we don't know if the organisms we're mating are homozygous or have the same 2 alleles in their system, or heterozygous, which means they have 2 different alleles in their system for the trait. And to make things more complicated, a trait may actually be controlled by more than 1 gene. So, selective breeding is a very, very long process of trial and error that occasionally results to some amazing accidents. However, considering overpopulation, climate change, and limited space, the agriculture industry is pressured into creating crops that can survive altered environments and produce more food with less resources. We do not have the time to do trial and error like our ancestors did. Despite the threats that they pose, our society seems not to be doing much to control overpopulation or mitigate climate change or even solve the COVID-19 problem. And our policymakers tend to be distracted by other intentions too. But going back to the science, the precise and accurate process of genetic engineering was invented so that we can create species and varieties of plants and animals that can solve some of these problems. The process of genetic modification is rooted on the fact that DNA is the blueprint for life. DNA is like the language of God encrypted in our cells that He has allowed us to decode and understand. Altering our DNA can change our looks, can change our abilities, and even heal some of our diseases. But it doesn't really change who we are. So, how does it work? Happening in your cell all the time is a process called transcription. Whenever your body needs a particular protein or enzymes, your cells communicate with each other so that they can produce what you need. Encrypted within your cells and within the DNA are discrete subunits called genes that contain instructions on how to make these needed proteins. In fact, only 1% of your total DNA contains these information-bearing genes. The rest of your DNA contain non-coding portions that controls how genes are expressed, such as the promoter region and the terminator region that indicate the start and end of a gene. Also, regulatory sequences around the genes can amplify or slow down the production of a particular protein. There are certain similarities between DNA replication and transcription, such as the location where the events occur, which is inside the nucleus, and the fact that the DNA double helix needs to be unzipped first before the events can occur. However, the similarities end there. And also, just as a point of clarification, DNA replication does not need to happen in order for transcription to occur. In fact, DNA replication only happens when the cells need to divide, while DNA transcription or the process of transcription happens all the time. The process of transcription begins with initiation. Here, the promoter region serves as the starting point and signal for the enzyme RNA polymerase to bind to the DNA template. Fun fact, in DNA replication, helicase is the enzyme that unwinds the DNA, but in the case of transcription, RNA polymerase does the job. RNA polymerase attaches free RNA nucleotides that complements the DNA template to create a temporary messenger molecule. Synthesis of the RNA molecule also occurs in a 5' to 3' direction. Elongation of this temporary RNA molecule ends when the RNA polymerase reaches the terminator region.
Hasta la vista, baby.
At this point, the newly synthesized RNA molecule and the RNA polymerase lets go of the DNA template. In prokaryotes, the newly produced messenger RNA can now directly proceed to the ribosome for protein synthesis. In eukaryotes, several other steps must still occur before the mRNA can leave the nucleus. Exons, the portions containing the encrypted instructions to make the protein, must be removed from the introns, the nonsense portion that lessens the chance that mutation will occur on the instruction-containing parts. Prior to this, a 5' cap and the poly-A tail is attached to the RNA molecule to protect it in its journey away from the nucleus. Once the splicing or removal of the introns are complete, the mature messenger RNA can leave the nucleus. And this ushers in the next stage, which is translation. Translation occurs in the cytoplasm of the cell in structures called the ribosome. And just like in the process of transcription, it has three main stages. initiation, elongation, and termination. During translation, mRNA nucleotide bases are read as codons of 3 bases as it is attached to the small subunit of the ribosome. tRNA molecules are responsible for bringing amino acids to the ribosome in the correct order, ready for polypeptide assembly. Each codon corresponds to a particular amino acid based on the genetic code as discovered by the team of Marshall-Nuremberg. In order for tRNA molecules to be replenished and carry free amino acids, amino acyl tRNA synthetases are charged and attach the corresponding free amino acid to the correct tRNA that bears the correct anticodon. For translation to begin, the start codon containing the sequence AUG must be recognized, and this is at the 5' end where the 5' cap is connected. This is a codon specific to the amino acid methionine, which is nearly always the first amino acid in a polypeptide chain. Now, at the 5' cap of the mRNA, the small 40S subunit of the ribosome binds. Subsequently, the large 60S subunit binds to complete the initiation complex. The next step, which is elongation, can now commence. In order to synthesize the protein, the ribosome translocates from the 5' end of the mRNA towards its 3' end, and the energy needed to create the protein is supplied through the molecule called GTP. Once the ribosome encounters one of the 3 stop codons possible, no tRNA molecule will bind to this codon, so the peptide and the tRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The mRNA then is subsequently destroyed. And voila! You have the protein needed by the cell in a matter of seconds to minutes. So, GMOs are created by tinkering with this process. Small snippets of DNA are introduced into an organism in the hopes of changing its physical characteristics. Examples of GMOs include plants that are resistant to pests, plants that are resistant to viruses, and plants that express essential vitamins. These traits are not artificial per se. They all come from nature, albeit a different organism. Since the mechanism of understanding the DNA is the same from a banana to a human, then little snippets of DNA that come from a different organism will be understood regardless of which organism you transfer it to. Also, genetic engineering does not entail the introduction of a toxin or a preservative into an organism. Genetic engineering just introduces a naturally occurring piece of DNA that will be destroyed with heat or enzymes found in our saliva upon consumption. Also, being a GMO is not contagious. Touching a GMO, being beside a GMO, or eating a GMO won't turn you or any other organism into a GMO yourself. But will GMOs make new GMOs just by existing? Security measures have been put in place to prevent this. Most GMOs have been engineered to be incapable of sexual reproduction. So, undesired reproduction or mixing with natural varieties are controlled. But won't GMOs result to reduced biodiversity? This is already an issue with traditional agriculture. And this is because we plant only the varieties that have enough nutrients and can grow fast enough to be sustainable to feed the current human population. In order for biodiversity to be threatened, we have to clear enough space that destroys the natural habitat of these natural varieties. And at the same time, the issue of survival of the fittest isn't really an issue because GMOs are not created to interbreed. So their lack of ability for sexual reproduction prevents them from competing at an evolutionary scale. But won't GMOs just make money for some and keep others poor? Though the capacity of GMOs to maximize limited resources have been proven, the fact that they can be patented by specific companies mean that one company can hold the monopoly for a particularly desired GMO. And this is where the issue of helping the environment, helping feed the people, can become an issue when the company doesn't want to be involved in those advocacies. Genetic engineering, just like the rest of the sciences, is a double-edged sword that can be used for good or for bad. It will always be in the intention of the user or the maker to justify whether the product is for good or for bad. So at the end of it, it is up to us if we want this technology to be used to help the least, the last, and the lost. I hope you were able to learn something from our short but hefty video for today on genetic modification. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipino scientist, in the comments section below. Don't forget to give this video a thumbs up and also to subscribe to our channel. Thank you very much and see you around!